Hello everyone and welcome to the fourth talk in our winter lunchtime lecture series on life and science. This season focuses on the human lives behind biological research. I'm Isabel Marina Held, Price Stone Postdoc Fellow at the Science History Institute. It's my honour today to introduce our speaker, Beans Veloci of Yale University. Before I do though, I'll say a bit about the format of today's lecture. Beans will speak for about 25 to 30 minutes, after which we'll have 15 minutes for audience Q&A. You can send in your questions during the talk using the chat function here on YouTube. You'll just need to log in and we'll be collecting the questions from the chat for the speaker. You can also email us your questions at lunchtimelectures@sciencehistory.org. That's lunchtimelectures@sciencehistory.org. Now I'd like to introduce our speaker for today. Beans Veloci is a PhD candidate in the Department of History at Yale University. They're currently finishing a dissertation titled Binary Logic, Race, Expertise, and the Persistence of Uncertainty in American Sex Research, which uses queer and feminist science and technology studies and histories of sexuality, race, and the life sciences to argue that sex works as a classification system because, quote, everyone has agreed that it doesn't have to make sense. Research for this talk was generously sponsored by fellowships from the American Philosophical Society and the Yale Fund for Lesbian and Gay Studies. Welcome, Beans, and thank you so much for joining us today.
Hello. So thanks everyone for being here, and thank you to the Science History Institute for inviting me to be part of this lecture series. So what I'm going to talk about today is a piece of one chapter of my dissertation, which is currently mercifully in its end stages. So the project as a whole looks at how sex researchers, broadly defined, working in the United States from the mid-19th through the mid-20th century, tried to sort the living world by sex. In the process, they staked their own claims to expertise on the special knowledges and techniques that they apparently possessed that enabled them to do this sorting work, and linked the facts that they were making about sex to the white supremacist social order that they desired to create. Underlying these efforts to organize a tidy tree of life, though, and this is the crux of the project, was a vast quantity of evidence that did not at all show an obvious or stable division between male and female bodies, or a singular thing called sex at all. A lot of the scholarship on the history of sex sciences, the history of sex science, describes the crystallization of categories and the increasing precision of definitions of sex around the turn of the 20th century. I focus instead on persistent ambiguities in the meaning of male and female and in the meaning of sex itself. I argue that sex amassed its power to sort bodies in this moment, not from solidified, agreed upon parameters, but a tacit agreement that it could be multiple, often contradictory things at once. Sex worked as a way of socially ordering the world with the cultural weight of science behind it, because researchers could classify bodies, redefine categorical criteria, and reconstitute what they considered sex at every turn. With this flexibility, unruly bodily forms that didn't fit into a binary could be recaptured back into normative categories of male and female, with no need to question a largely binary sex system or the right of science to serve as a privileged site of sexual knowledge. One of the major sites where that was happening was eugenics. And there's a whole story that I can tell about the relationship between sexology and eugenics, and the way that anxieties about eugenic sterilization causing sex transformations got kind of swept under the rug, which for that stuff, keep your eyes peeled for a book in like 5 years from now, or ask me about it in half an hour. Today, what I'm going to tell you about is the research side of eugenics and how, in the service of racial order, a lot of it made a mess of what sex was. In the early years of the 20th century, researchers at the Eugenics Record Office and the Station for Experimental Evolution in Cold Spring Harbor, New York, dreamt of achieving white perfection through science. As they set out to understand heredity and gain control over reproduction, much of their research came to be about sex and its role in making better forms of life. So this talk explores the way that eugenics scientists deployed a really capacious understanding of what sex was, depending on what species they were using in their research and what kinds of problems they wanted to solve. For eugenic researchers, sex was not a pre-existing unified object of study, but rather something they actively made into a constellation of traits and processes. While something called sex sometimes functioned as a binary variable of male or female used for making sense of vast quantities of eugenic research data, sex could also be malleable bodily morphology, reproductive capacity, hormonal makeup, metabolism, and behavior. Eugenicists in the early 20th century depended on this multiplicity of sexes in order to make their claims to scientifically backed racial authority work. So both the Eugenics Record Office, or ERO, and the Station for Experimental Evolution, or SEE, were founded by eugenicist and biologist Charles Davenport, whose name might ring a bell if you were here for Geneton's lecture a few weeks ago, where he was one of the scientists working in the masculine world of turn-of-the-century zoology. Davenport initially established the Station for Experimental Evolution in 1904, with the goal of using new experimental methods to learn about species development and the inheritance of traits, and to try to recreate Darwinian selection by artificial means in a lab environment. But over the next several years, Davenport became interested in the inheritance of not only physical, but psychological and behavioral traits in humans. Thanks to the influence of his wife Gertrude, who was a prolific eugenic researcher in her own right, as well as through his involvement in the American Breeders' Association Eugenics Committee, Davenport decided to put these interests together and use the SEE as a space to figure out how to breed for the best human traits by using study of animals, and figure out how to eradicate traits that in humans led to things like, or supposedly led to things like crime, economic waste, and other forms of social degeneration. So eugenics became the primary focus of research at the SEE. In 1910, Davenport secured additional funding to create the Eugenics Record Office, whose staff aimed to fully document the inheritance of various traits in the American population by amassing a truly ridiculous quantity of family histories, which we'll get to in a minute. So Cold Spring Harbor quickly became an epicenter of eugenic research in the United States. Eugenics was ultimately a matter of breeding, of controlling the future by controlling sexual activity and sexed bodies in the present. And it could not function without a thorough understanding of reproduction, sexual behavior, and sex itself. And so sex was built into every aspect of eugenic research at Cold Spring Harbor, but it was built in very different ways. So at the SEE, researchers emphasized scientific control over sexual malleability and investigated the wide range of forms that sex could take across the animal and plant kingdoms. At the ERO, data collection and analysis depended on a fixed binary model of sex that was useful for tracking heredity of traits and making copious amounts of information both sortable and intelligible. The coexistence of these approaches leads me to the argument that making racial claims based on sex and trying to make biological racial changes based on sex required a flexible meaning of sex itself. The jumble of meanings and uses of sex that persists in science today has one of its origins in this eugenic history. So let's start with the Station for Experimental Evolution. The SEE focused on research in animals, but Director Charles Davenport was explicit about his desires that investigations in rats, pigeons, and even fungi be transferable to human eugenics. Quote, "Neither physically nor physiologically is there a sharp break in the animal series where it culminates in man," Davenport reflected in a 1907 report on the SEE's doings. Consequently, he continued, the discovery of the laws of organic evolution is a study of human evolution. Since when we know the law, we may control the process, the principles of evolution will show the way to an improvement of the human race. So the study of the laws of heredity and evolution that Davenport so valued quickly led SEE researchers to the study of sex because, as he put it, the study of sex was fundamental to, quote, discovering the essential nature of the living stuff. Sex research was so prevalent in the SEE's labs that by 1925, Davenport was characterizing the SEE as, quote, devoted to a study of the phenomena of reproduction and development in animals and plants. Whereas he told the eugenicist, gynecologist, and sexologist Robert Latu Dickinson, who's the letter that he says this in, you can see here in kind of blurry text. For many years, SEE researchers had been working on topics like, and this is the list here, sex differentiation with special reference to its basis in the chromosomes, the basis of the ability to call forth male production in a parthenogenetic species, the phenomenon of exclusive male production in certain matings, control of primary and secondary sex characters in pigeons, the chemical basis of sex distinction where there is no morphological distinction, and the study of sex intergrades and their hereditary basis. In this letter to Dickinson, Davenport again emphasized that this animal research was of human relevance. We have no doubt at all, he concluded, that general principles secured from a study of the lower organisms will be applicable to man. The outcome of all of this research was a version of sex that was not only incredibly variable in nature, but also one that could be modified in the lab by expert hands. Transforming sex characteristics in poultry was a topic of keen interest to SEE researchers, in an effort to figure out what exactly determines sex in an individual and how that be controlled through breeding practices. So, for example, in 1912, Hubert Dana Goodale, at the time a graduate student working on genetics at the SEE, removed the left ovary of a female chicken that subsequently assumed, quote, most but not all of the characters of the male. The experiment led to tremendous excitement on Davenport's part because it, quote, seems well calculated to demonstrate the essentially hermaphrodite nature of the female fowl, and thus the possibility that sex could be modified. To Goodale, the experiment indicated that scientists, quote, need no longer erect separate categories for characters that appear in one sex only, but may classify a given character in the female with the corresponding character in the male. That is to say, particular feather patterns, to use one of Goodale's examples, were not actually male or female patterns. The determining factor, as Goodale put it, is not in itself sex, but other bodily processes like the circulation of internal secretions, as he called them, which again gave evidence to the possibility of editing an individual body to improve its traits. Goodale's experiments with avian gonadectomies demonstrated that experimental expertise could be wielded in order to modify characteristics in sexually malleable animal bodies. So Goodale would soon depart the SEE, but by 1916, other work on sex at the SEE enabled Davenport to make even bolder statements about how sex actually functioned. To the station, Davenport wrote in that year's annual report, seems to have fallen the opportunity of demonstrating that the current view that sex is determined solely by the sex chromosome is too narrow. Davenport based this claim primarily on the work of Oscar Riddle, who held a position as a research associate at the SEE from 1912 until his retirement in 1945. Riddle spent his career developing a metabolic theory of sex, which held that sex was plastic and subject to changes in caloric use. With this theory informing his research, Riddle argued that he could determine the sex of future offspring through particular breeding practices, thus controlling male and female sex ratios. But perhaps most thrilling to Davenport was Riddle's work on what he called sex reversal in pigeons, the transformation of male pigeons into female ones and vice versa. Through injections of testicular and ovarian extracts, Riddle was able to change the sex behavior and secondary sex characteristics of his experimental birds. Riddle's work, along with that of his colleagues like Goodale and others working on what they called sex intergrades, led Davenport to the conclusion that, quote, sex in general is a much less fixed and precise state than is commonly supposed. Eugenic science could use that to its advantage. As Riddle put it, quote, the demonstration of the complete transformability of one truly hereditary and chromosome-determined character, that is, sex, now makes it quite illogical to assume that any hereditary character similarly founded in the germ may not be transformed in and during the life of the individual. This aspect of our knowledge of the heredity and development, when adequately developed and applied, should prove of much interest and of very real practical value to mankind. So, some SEE research demonstrated that sex was not a static or binary thing. Other research expanded the definition of sex itself, and perhaps the best example of that is the work of botanist Albert Blakesley and Sophia Cetina on sex in fungi. Of course, none of the familiar markers of sex existed in fungi. There were no genitals, gonads, or secondary sex characteristics to even begin to attempt to sort out or begin modifying. Blakesley and Cetina had to decide what would even count as sex. So Blakesley had been working on the problem of sex in mucors, a genus of molds, since at least the early 1910s. In these early experiments, Blakesley sought to figure out which of the, quote, sexual races of the mucors, which at that point were simply referred to as plus or minus, were in fact male and female. This was a difficult task because they were, quote, nearly indistinguishable from each other. Ultimately, Blakesley determined that the, quote, vegetatively more luxuriant race may be considered female, while the less luxuriant fungi were male. Blakesley had come to this conclusion by demonstrating what he called a sexual reaction between these fungi of differing luxuriances with gametes of two types that he had defined either as male because they were smaller or female because they were larger. So for Blakesley, sex was not malleable in quite the same way that it was for Goodale and Riddle. Blakesley was more interested in sorting fungi into stable sex categories than demonstrating that sex could be modified in the lab. Yet, Blakesley's work demanded its own version of sex itself, thanks to the species upon which he performed his research. In the world of fungi, sex was gamete size, vegetative luxuriance, and as he and Cetina would later come to claim, based in chemical difference. The issue here was not whether sex could be changed, but what sex even was. Questions of how to determine and understand sex in fungi would continue to interest Blakesley through the 1920s. In those years, he was joined at the SEE, which was by then a subset of the Carnegie Institution of Washington Department of Genetics, by research associate Sophie Cetina. Together, they continued working on the problem of how to identify sex in fungi, or as their initial proposal to Davenport put it, quote, they would test the biochemical differences between the cell contents of males and females. To tease out these details, they conducted a variety of tests at the microscopic level. So instead of looking for vegetative luxuriance, as Blakesley had done earlier in his career, Blakesley and Cetina ground up their mucors, applied alcohol or distilled water, and centrifuged them, winding up with a liquid extract that they could perform their tests on. So unlike in sex research being conducted on birds or mammals, the body of the subject was not needed to make a determination about sex. So extract in hand, Blakesley and Cetina conducted a wide range of tests that they hoped could identify sex according to the smallest markers. They looked for the presence of various enzymes in the extracts and how much of the extract was required to oxidize a sample of potassium permanganate. They tested the pH of the extracts. They assessed individual gametes for the presence of a yellow substance that they presumed to be fat and gauged the gametes' reactions with tellurium salts. The outcomes of these various tests matched what Blakesley had determined over a decade previously, that in their biochemical reactions, quote, The plus sex of mucors corresponds to the female, and the minus sex corresponds to the male. The details here are not super important. The point that I want to emphasize is that when Blakesley and Satina decided what their markers for sex would be, they were vastly different markers than what Goodale and Riddle would have used. Sex for Blakesley and Satina could be abstracted from a classification system in which there were certain kinds of bodies or bodily characteristics at all, and reduced to a series of male and female chemical reactions. So at the SEE, what sex was varied. Sex was malleable there, not just in the sense that female pigeons could be transformed into male ones, but in that sex itself could be different things for different research purposes. It could be the presence or absence of certain body parts, of chemical reactions, of different kinds of hormones, and it could be an attribute of a body that could be changed or a fixed state to be identified. And these different enactments of sex were happening concurrently. Next door, at the Eugenics Record Office, sex research took yet another form. As Albert Blakesley framed it after he took over as director of the SEE upon Davenport's retirement, quote, "Matings cannot be controlled in man as in other genetic subjects. One could not experimentally breed humans and see what happened." So the ERO's mission was to trace the results of human matings that had already occurred by collecting and organizing data about human heredity with the express purpose of using that information to eugenic ends. The ERO chased these goals in part by accumulating its own library, hiring researchers to comb through medical journals and other potentially useful literature, and publishing pamphlets, books, and a journal called the Eugenical News. But its primary research method was the collection of family histories, partly through the voluntary donation of information by people who wanted to contribute to eugenic science or participate in fitter family contests, and partly through extractive research based on largely incarcerated or otherwise institutionalized people. Between 1910 and 1924, the ERO trained a corps of 258 field workers, most of them women who had a background, sometimes at the doctoral level, in biology or medicine. These field workers would travel to institutions like Kings Park State Hospital for the Insane, the Massachusetts State Industrial School for Girls, and Sing Sing Prison, and to communities known to have particularly large and distinctive families or isolated populations. In those spaces, through a combination of interviews and combing institutional records, they recorded extensive individual and family histories with an eye for anything that might be a heritable trait. The ERO also teamed up with other institutions, independent volunteers, and the American Eugenics Society's Fitter Family contests to accumulate information beyond the reach of its own field workers. The result was a massive collection of pedigree charts analyzed for which traits could be passed down. So, as we know, Davenport, who led the ERO along with Superintendent Harry H. Lawton, had a vested interest in sex. At the SEE, whereas at the SEE, that interest suffused almost every aspect of the research that took place at the ERO. Davenport was, like many of his contemporaries, concerned about the social impacts of what he perceived as sexual immorality, and ERO fieldworkers' attention to prostitution, extramarital sex, and sodomy in their reports and pedigree charts in part reflects these concerns. Fieldworkers also investigated sexual aspects of the body and their effects on health, especially regarding menstruation and sexual development. The fieldworkers were on the lookout for dysgenic forms of sexual behavior and reproductive trouble, and they also uncovered a fair share of instances of cross-dressing or ambiguously sexed bodies. For the most part, though, unlike their colleagues at the SEE, those at the ERO spent little, if any, time hashing out the meanings of maleness or femaleness, or struggling to sort the two categories out, or thinking about how one might go from one to the other. On the contrary, the ERO's entire method of collecting and making sense of data relied on a static binary model of sex. So let me pause for a second to make clear exactly how much data they had. By the time the ERO shut its doors in 1930, it had accumulated nearly 1 million index cards that consisted of perhaps 35,000 individual records. This was a small subset of the mass of paper that constituted the ERO's records, which further included endless stacks of multi-page record of family traits forms that organized information about traits that seemed to crop up among individual families, and pedigree charts, often multiple sheets of paper fastened together to form a multi-foot record that the field workers made to visually represent the data that they collected as they interviewed subjects. When the ERO shipped its records to the Dyke Institute for the Promotion of Human Genetics for safekeeping in 1948, this is one of my favorite things that I learned in this process, the receipts from their moving company indicated that they shipped 18,230 pounds of materials. And that was like after getting rid of a bunch of stuff. So for decades, the ERO had to organize and make sense of all of this information, and they turned to sex to do so. The basic unit of data collection that filled their filing cabinets was the family history. I'm going to do a quick dive into the actual material form that these histories took, because paperwork is never merely paperwork. Any blank form to be filled in or guidelines of how to collect data places normative constraints on what information can be collected and bounds what can be considered possible. In this case, a binary model of sex was their way of sorting people. So for those who wanted to donate their family histories to eugenic science, whether students in genetics classes, members of eugenics organizations, or friends and family of field workers, the ERO had a set of standardized pre-printed family history forms that interested parties could have mailed to them, fill in at their leisure, and send back for processing. So the abridged record of family traits, which you can see a page of here, was one of them. These forms were organized by family role, which meant that every single piece of information that the ERO collected about these families was filtered through normatively sexed family and reproductive relations. One could not fill in the form without explicitly classifying each family member by sex through their family relationship to the person inputting information into the form. While each form allowed the person filling it out to choose which traits to focus on, depending on what they thought was most common among their family members, the way that they had to organize that information was not up to them. The goal was specifically to track traits through reproductive family lineages, which made binarily sexed breeding pairs the focal point, such that it was not even possible to voluntarily send in information beyond using the provided form. Sex was equally important for representing data in the form of pedigree charts. So these charts allowed for the visualization of an entire family through multiple generations, with specific kinds of notation to indicate whether or not a family member possessed a certain trait. The entire structure of the pedigree chart relied on sex. So if you take a look here at the ERO's brief instructions for making a scientific family study, which was located inside the cover of the family distribution of personal traits form, you'll find that binary sex is actually crucial for organizing one's family data. The instructions provide a template for turning all of the individual records into a pedigree chart for the entire family. And the first item listed in the key to the sample chart symbols is that circle equals female and square equals male. And this was not just the voluntary form. This was how all family histories were recorded on pedigree charts with these squares and circles. So pedigree charts emphasize sex not just relationally, but as the core attribute of an individual person rather than a mere descriptor. So in the ERO's very early days, each person on a given pedigree was represented by a circle, and sex was noted next to the circle alongside any other traits. The ERO quickly turned, though, to a method in which the circle and accompanying sex annotation was replaced by the square for male and the circle for female to represent each person. The individual, in effect, became their sex. Furthermore, each chart had to be hand-drawn, And while some of the field workers had square and circle stamps to do that aspect of chart drawing for them, others carefully penned in each individual square and circle. Even those who had the stamps at their disposal would have had to continually switch back and forth between the two stamps, using the tools for each and every individual that they recorded on a pedigree, which could number over 100 in a single family chart. So the kinds of practices engaged in by ERO field workers wind up reinforcing a binary model as people filled out forms, as eugenicists painstakingly drew circles and squares in their pedigrees, and in the conversations that field workers had with their research subjects. That repetition, and particularly the way that ERO field workers brought these expert perspectives into non-expert communities, probably helped spread and perpetuate the idea that science backed a singular binary understanding of sex, even as that was only one facet of how sex worked in the eugenics labs at Cold Spring Harbor. Recall, though, that in addition to his role at the ERO, Charles Davenport, the one who was making the decisions about these notation systems, served as director of the SEE. He was regularly in close contact with and received detailed reports on all projects being conducted from researchers like Hubert Dana Goodale and Oscar Riddle and Albert Blakesley, whose work actively argued against the existence of stable binary sex, or at least called into question how easy it might be to identify. And Davenport clearly supported that work. Otherwise, he likely wouldn't have pushed for its being funded, nor hired and kept on staff the researchers who were doing it. Yet, none of these ideas made it into the ERO's data collection or organization methods, which relied entirely on the assumption that humans were split into obvious categories of male and female. So the mismatch between the complex model of sex used at the SEE and the binary stable one used at the ERO could, if one were so inclined, be chalked up to a distinction between non-human and human research. Maybe it was okay for pigeon sex to be malleable if the assumption was that that had nothing to do with people. But as other scholars have shown, in this very moment, scientists were using animal research to try to modify sex characteristics in humans. Even at the SEE, the point was that animal research would be translatable to human eugenic policy. Meanwhile, as I've written elsewhere, a fluid spectrum of sex was routinely deployed in the decades leading up to this research to make arguments for the supposed evolutionary superiority of whiteness on the basis that only white people had achieved full sexual dimorphism, while less advanced races had more in common with hermaphroditic animals. So what is going on? When seen through the lens of eugenics, sex multiplies. At Cold Spring Harbor, sex was a trait that an individual could have, a behavior an individual could engage in, and a classification system for organizing research. It was a fixed designation as male or female, and also a malleable aspect of the body subject to environmental, surgical, and metabolic influences. Sex was not a single thing, but an object differently enacted depending on the particular research questions and methods at hand. And these disparate meanings of sex did not have to coalesce into one. On the contrary, the eugenic research that took place at Cold Spring Harbor was able to take place because multiple contradictory sexes existed at the same time. A complicated version of sex was perfectly well and good in the labs of the SEE. In that context, scientists' ability to modify sex characteristics in their experiments or identify sex imagined as an invisible trait manifested through chemical reactions emphasized the power of the power of science over bodies. In the abstract, more scientific control over sex, regardless of exactly what sex meant, was good for eugenics because it allowed researchers to imagine a world in which they could have more control over breeding. But when it came to the collecting and sorting of thousands upon thousands of family histories, the SEE's enactment of sex simply would not do. This wasn't an instance of binary sex being taken for granted. The so-called nature of sex was under investigation mere minutes walk away by people who assumed sex not to be binary. The knowledge about malleable sex that they were producing, however, turned out to be inconvenient for eugenic research and practice. How would you carry out family history data organization or draw a pedigree chart with a more nuanced understanding of sex? How would you justify pushing for mass campaigns of sterilization if you take seriously the work coming out of your own labs about how sex can actually be changed in an individual body? Different methods of solving racial problems with science, as the eugenicists would have it, required different sexes. Scientists at Cold Spring Harbor did not have a consistent or coherent or unified understanding of sex. And this was not a problem for the people doing research there. Rather, sex functioned as an umbrella category for things that clumped together in frequently contradictory ways. The body, the mind, reproduction, behavior, hormones, gametes, morality, family role, a square or circle on form. What mattered was what would best serve the eugenic mission. And that's ultimately the main takeaway here. This is not just me poking fun at early 20th century scientists for being inconsistent. It's an important part of the origin story of 20th and 21st century sex science, where sex as a scientific concept developed to contain multiplicity so that it could be deployed in a whole range of ways in the service of racial violence. So what I leave you with is this. There is not a pre-existing thing called sex whose boundaries can be mapped, whether by researchers in the past or historians in the present. Only something made by a set of research practices responding, in the case of eugenics, to a desire for white domination. Thank you.
Thank you for your talk, Beans. It was a really great talk, and let's see if there's any comments, any questions in the comments yet. Okay. So let's see if there'll be any questions coming up. I was really blown away by your talk and was really interested in how the two institutions were geographically so close to each other. When did you find that out in your research?
Well, so there's in some ways a kind of funny story, as funny as a story about this location can be. I actually went to summer camp as like a young teenager at Cold Spring Harbor Laboratory. And like there's a genetics camp for nerdy children that I went to. And so I actually had a very good sense of the space before I even knew what was happening there. And so one of the reasons that I got into this particular topic was a little bit accidental, in that I kept stumbling across Cold Spring Harbor stuff, and I was like, wait, I have been there. I know exactly where they're talking about. And so that was one of the things that actually led me to really think about, like, how are these two things going on at the same time, when they're literally like you walk down a little street from like one building to the next. And that kind of got me to start thinking about how it is that they have these massively different frameworks, even as they're so close together.
Yeah, it's really interesting to think about in general in historical research about these sorts of missed opportunities in a way, like what would have happened if things had been different in a way, and that there had been kind of more sort of more of an energy into like thinking about sex as malleable, also not just for non-humans but also for humans, and to think about the kind of I guess the way is that they weren't allowing that communication to take place.
Yeah, and I think, I mean, speaks to, you know, contemporary university campuses right now, or like, I mean, in some places there are like massive spatial gulfs between, you know, what's happening in the sciences and what is happening in, you know, a gender studies department. But so we're, I think, still kind of encountering some of the same ways that information can be made in close proximity without the actual people really figuring out ways to communicate with each other. Although in this case, I mean, I would say that like there's definitely like there are like trans studies scholars right now who are kind of starting to put together the ways that like eugenic theories of sexual malleability end up producing some of the possibilities for even thinking about transforming human bodies that leads to trans people being able to medically transition and kind of grappling with that history and like what does that mean in a contemporary sense of like thinking about sexual malleability as something that might be liberating, but at the same time it has this incredibly violent past, and kind of what do we do with that?
Yeah, that's really exciting. Let me just see, we've got some questions coming in now. Pratima says, how these sources treat sex as malleable. So the question is relating to how these sources treat sex as malleable. I was wondering, did the eugenicists you're talking aspire to make sex into the kind of universal category it's become, or did that just sort of happen?
Yeah, that's a good question. That like, I mean, yeah, yes and no. I mean, I think a lot of where like the kind of expertise part of my project that is like alluded to in the subtitle of my dissertation is that a lot of scientific expertise comes out of these not being fully universal theories. That one of the ways that people position their own research as important is by kind of saying like, I am the only one who can solve this problem with a particular method, with a particular test subject. And so that kind of, you know, they almost don't want these like their work to become universally applicable, because otherwise it makes them, you know, how are they like going to get funding for it if it's totally universal? Which again, like a contemporary project is this kind of need to articulate your own kind of like heroic status, I guess, in doing your research. At the same time, I mean, this kind of also gets into the ways that sexology and eugenics are really entangled at this moment, is that like a lot of the funding for the SEE and some of the funding for the ERO as well comes from the National Committee for, I always mix this up, Problems in Sex Research, I think is the acronym, where it's the hope is that this like the work that's being done in eugenic settings is something that can be used to illuminate topics of sex as a whole. So it's a little bit of both.
Great, thank you. We've got there's been some really positive reactions to the talk. It was so informative. So H. Brown says, again, this was so much new information for me. Thank you for your amazing talk. What do you think are some future applications for your research, and how would you hope it's used?
Yeah, I'm doing some really exciting work with scientist colleagues, I think some of whom may be listening in on this, where we're trying to kind of see how this works by this, I mean the kind of like multitudinous definition of sex, or multiple definitions of sex that are being used in scientific research, plays out in a contemporary sense. Because this way that science gets used, that sex gets used to refer to totally different things, but all kind of wrapped up in this one category, that's just without really any consideration of like how those different components actually come to be like seen as related to each other is something that still happens in science. So I'm hoping that this kind of historical perspective will ultimately be useful for thinking about how people doing sex research, or just any research that uses sex as a research variable, can think a little bit more critically about that category, and hopefully naturalize binary sex a little bit less.
Yeah, I think it's really useful in that way to sort of revisit when these ideas that we're still living with the legacies with today and where they come from. Okay, so we have another question from Ruth Rand, who is a postdoc at SHI. And she says also, thank you for the talk. I wondered if you might share how the material you share today is positioned within your larger dissertation or in 5 years book project, which we're obviously very excited for.
Great, thank you. Yeah, I'm seeing another question come up after this about kind of how I found my way through the archives, and I'm going to answer these questions a little bit together because it's kind of I think a lot of the same answer to those. So this particular part of the dissertation kind of comes in the middle. So I start with looking at kind of pre-eugenics or pre-formal eugenics animal studies, mostly in zoology, looking at how scientists were kind of trying to make arguments about racial difference based on this kind of like supposed differential of sexual dimorphism that I kind of talked about very briefly, where whiteness is kind of the pinnacle of sexual dimorphism, and the way that you can detect racial primitivity is by apparently less sexual dimorphism. So start there, move into eugenics, and then from there, this is kind of where it gets into the how did I make my way through the archives, which was this kind of following the breadcrumbs kind of situation of realizing that, so in the first chapter, a lot of the animal people were doing agricultural stuff. That led me to like the American Breeders Association eugenics people, which led me to these eugenics people. So there's this kind of breeding aspect of it that links the two ways. And then Robert Latu Dickinson, who is a correspondent of Davenport that I mentioned in this talk, is the focus of the next chapter, which looks at kind of in a similar way, like gynecological models of sex, where on one hand you have someone like Dickinson, who is this kind of massive figure in the field, who's in touch with these eugenicists, and so is kind of like taking up their very malleable model of sex to make claims about how humans are not actually a binary. But then if you look at that gynecologist like clinical notes and clinical practice, similarly to this kind of disjuncture between what the SEE and ERO are doing, you have in gynecology a kind of similar thing where on a theoretical level, you have this very expansive understanding of what sex looks like. And then on the kind of everyday practical level, no one ever gets ejected from the category of woman, no matter what their body is actually doing. There was going to be another chapter on like statistical research methods that, thanks to the pandemic, it's not happening. And then I end with the kind of development of trans medicine and the kind of shift from male-female being the kind of sex boundary that's in question to cis and trans being the sex boundary that's in question. So these are all kind of like, but among them and between them is a trajectory of people who know each other, who are talking to each other, who are on the same conference panels, who are summering at each other's beach homes, all kind of trading back and forth these different models of how sex works. And in the process, kind of all tacitly coming to the agreement that these are all valid models and they don't have to kind of figure out which one is the right one, because if they figured out which one is the right one, none of them would be able to do their work.
Yeah, I think that sort of ties in with your great quote that you have that everyone agreed that it doesn't have to make sense, right?
Yeah, and like it was a little bit like facetious when I started describing the project that way, but I think that really is kind of ultimately what it comes down to is that the only way that science is able to kind of maintain its grip on being the privileged site of knowledge production about sex is by everyone just kind of being like, well, it can be different things in different places, and that's okay. Which is a very, very different, people who tend to use kind of like science as, supposedly saying that like binary sex is like biological or whatever, are kind of trotting out this science in the abstract as a kind of cultural producer, in a way that is very, very separate from what science itself has said for, the past over 100 years. Which is not to say that like this eugenic model of sexual malleability is the road to justice does not lie in eugenics. Like that is decidedly not what is going on. But what I'm kind of trying to point to is the way that science does much more. It's much more coherent as a kind of cultural knowledge producer, or like a kind of like cultural figure as a knowledge producer about sex than it is in actuality.
Yeah, I think something that really came across in your talk as well is the kind of concept of there are gatekeepers who determine what sex is, and also who get to determine when it can and can't make sense. Do you know what I mean?
Yeah, I mean, and I think like this is still like continues, to be a contemporary issue where, I think it was like 2 years ago that like the US federal government was like, we're gonna just like declare that sex is binary because that's what science says. And that kind of move towards what science says is kind of its own form of intellectual gatekeeping, in a way that assumes that science can't possibly be allied with queerness, or be allied with even kind of an imprecision almost. Like there's in that kind of cultural imagining of science that is used to really like violent transphobic political ends is so disjointed from what a lot of from how things could be otherwise in science. And so I mean one of the things that I end up talking about a lot is like the thing here is not to be like, oh, we therefore need to do science better and be much more precise about what we mean by sex, but actually we need to question why it is that science is supposedly the be all and end all of what sex or what gender is, and start to think a little bit more expansively about how scholarship in other areas, how lived experience in other areas are equally, if not sometimes more legitimate forms of knowledge in those areas.
Yeah. I had one more question as well. I really liked how you talked about that you liked that you found the fact that the ERO had over £18,000 worth of material in 1948. You kind of pulled out that fact at one point, and I was just wondering, because you went to the archives and you used the materials, did you have all the materials still intact from that period, or have they been edited down in some way, or is that just a snapshot that hasn't changed?
So they've been the documents have moved around a lot since then. So they're now, most of them are at the American Philosophical Society, but as I mentioned, like they were originally moved to the Dite Institute, and then at some point the like LDS church wound up getting a bunch of them because of the like genealogy research aspects of it. So they were actually the ones who like microfilmed a bunch. So anyway, what I wound up looking at was like I don't think the APS has all of it, but it's certainly a lot. I spent like almost an entire month just even kind of skimming the surface of what was in the field worker records. They're just like it's just an incredible kind of spooky quantity. I mean, there's, I'm not a particularly wooey person, but there was something about just like going through box after box after box of like, records that had often come out of prisons and psychiatric institutions, where you just really like, out of all of the archival work I've done, it was the one where I just like the vast quantity just really felt weird to me and felt uncomfortable, which is I think like is one of the reasons that like, I put that image of all the filing cabinets in there, and that I'm so interested by this question of like the mass of information, because it just speaks to like the immense power and reach of this institution, that they just went through the effort of trying to distill so many lives into paperwork, and the kind of bureaucratic violence aspect of it is something that is, like interesting is one way to put it, but also just like alarming. So, but yes, it did take me a long time to go through a lot of paper.
I can imagine. And then finally, just out of curiosity, what did you get up to at genetics summer camp?
It was a lot of pipetting. Okay. That was we did we made fruit flies glow in the dark. That's my big memory of that, is pipetting and being in, we like did our experiments over the course of a couple of weeks, and then took our little fruit flies in their little containers into a dark auditorium, and they like turned the lights off, and then they were glowing.
That sounds great. I like the way that you talked about kind of realizing that you'd just been down the road and it all kind of coming together in a way in your research.
It was a fun is not the right word for like this topic, but like it's always I think it kind of helps with the like historical imagination when you've physically been in a place.
Well, Beans, thank you so much. That was a really fantastic talk. I think we've run out of time for questions. So again, thank you. This talk has been the fourth of 6 in this winter series. We're streaming every Wednesday at 1 p.m. Eastern Time. Next week we have Emily Herring of Leeds University, who will give the next talk. This is entitled Henry Bergson's Creative Evolution and 20th Century Biology. So please do join us for that next Wednesday. That's March the 17th at 1 p.m. Eastern Time. And for more from the Science History Institute, including updates on all our virtual programs and events, you can follow us online. We're at sciencehistory.org on Twitter, Facebook, and Instagram. Thank you so much again for joining us. Have a great rest of your day, and we'll see you next week.